All right, the sooner we get done, the sooner we can eat lunch, right? Okay, so we're going to talk about smooth muscle now. Parts of this lecture are a nice segue from the endothelial cell lecture. I'll tie a few things in. Some of it might be a little bit of a review from what you've learned about skeletal and cardiac muscle, and some of this will be brand new. So you can see your objectives. We're going to start off with this.
Smooth muscle tissue is in the walls of hollow organs throughout the body. The tissue contracts and relaxes to contain substances and move substances through the body. Smooth muscle contractions are involuntary actions managed by impulses that travel through the autonomic nervous system to the smooth muscle tissue. The arrangement of cells in smooth muscle tissue provides for contraction and relaxation with great elasticity. The smooth muscle in the digestive tract contracts and relaxes in peristaltic waves that move swallowed food and nutrients from the mouth through the stomach and the intestines. Artery walls include smooth muscle that relaxes and contracts as pressure changes move changing volumes of blood through the vessel. The smooth muscle of the bladder and uterus allow for great elasticity and contraction of those organs.
Okay, we're done. We can go. Just kidding. Okay, so that's a nice overview of smooth muscle. Smooth muscle is really unique. There are three muscle types, skeletal, cardiac, and smooth. Why is smooth muscle so really unique? You need a muscle that can contract for the majority of the day, whether you're awake, asleep, unconscious. Okay? Because it's contracting for the majority of the day, it needs to be able to do that very efficiently in terms of energy usage. You need a muscle that can mix the contents of a lumen even when the muscle is already stretched out. You need a muscle that can maintain the shape of an organ even when there's no bone for that muscle to attach to. Think of your bladder. We need a muscle that can respond to a local influence as well as neurohormonal influence, as well as the brain. Because of the constant contraction, this muscle has to be very energy efficient. So smooth muscle is really unique. So let's talk about some general characteristics. It's slightly different than endothelial cells. Smooth muscle is also heterogeneous, but there's really only two flavors. There's a multi-unit smooth muscle. It's composed of discrete, independently innervated nerve fibers. So every muscle cell has its own synapse. It's controlled by nerve signals, as if every muscle cell had its own motor unit. Single unit you also see referred to as unitary, found in the walls of the gut and the arteries. It's not characterized by neuromuscular junctions. But what characterizes single unit smooth muscle are gap junctions between the cells. And I know you've learned about gap junctions. Gap junctions allow electrical activity, ion flux, to flow between cells in a uniform fashion, as if that sheet of muscle cells behaved as one motor unit. Okay? Single unit is what we'll really talk about today. It's arranged in sheets and bundles so that cell membranes are always in contact with each other. They have gap junctions so that ions can flow freely, and you have electrical activity that can allow this sheet of muscle cells to act in unison. And when you think about it, that has to be. If we were talking about the gut, they all have to be coordinated in their contraction to squeeze material through in a directional manner. If we're talking about an artery and needing to maintain blood pressure through lumen diameter, every smooth muscle cell in that segment of artery needs to be on the same page. So this is a cartoon of what I was just talking about. An example of where multi-unit smooth muscle cells are in the iris of the eye. Every cell has a junction. Single unit, there may be one or two junctions, but what I really want you to know is gap junctions permit coordinated contraction. Viscera, just about every blood vessel you have. We can learn a little bit about smooth muscle by comparing it with skeletal or striated muscle. So you've looked under a microscope as undergrads or now at striated muscle, and it's very distinguishable appearance. there are bands and zones, A band, Z discs, and all those kind of things. We all looked at them as undergrads. Smooth muscle is different. They have actin and myosin, but it's not organized in a sarcomere. They're very loosely organized, thick filaments, which is myosin, and thin filaments, which is actin. So when you look at a smooth muscle cell under a microscope, it looks like a fibroblast. You do not see sarcomeres and this defined structure like you do in cardiac and skeletal. In a smooth muscle cell, in contrast, the actin and myosin are, the actin actually is attached. So the actin on one end is attached to a structure in the cytoplasm called a dense body. The other end of the actin is attached to a structure called an attachment plaque. The attachment plaques are found directly on the inside of the plasma membrane. So one side of the strand is attached to the cytoplasm, to a structure. The other side is attached to the membrane, to another structure called an attachment plaque. That allows the smooth muscle to contract. And when we go into the actual mechanism, you'll see how that works. Smooth muscles can contract in response to either neuromuscular synaptic transmission or else electrical coupling that's local. And we'll talk about that in a minute. The real important thing to think about is that smooth muscle requires much, much less energy to contract and maintain that contraction compared with striated muscle. So this is a really nice graph. So this will be skeletal muscle, this is cardiac muscle, this axis is tension, and this is time. And you can see it's not even close. Smooth muscle cells are much slower in developing tension, but they can maintain that tension for much longer periods of time without fatiguing. And it allows the walls of organs to maintain this tension without exhausting your supply of ATP. And the velocity of contraction is much slower as well, too. When we talk about calcium mobilization, we'll get into the molecular mechanisms. So do an experiment right now. In your chairs, do a curl and push against yourself as hard as you can. Keep going. And I don't care how many times a week you go to the gym, within about a minute, maybe 2 minutes, you're going to feel your muscle fatigue, right? That's the only picture of a bladder I could find. Smooth muscle is different. Okay? So think about it. If you started your day with one of those 20-ounce Wawa coffees, or now it's 11, it's finding your way into your bladder. What if the smooth muscle cell that maintains the shape of your bladder utilized energy the same way as your biceps? You'd be exhausted. You'd have to run to the restroom every 5 minutes. Or if any of the ladies in here ever gave birth, the placenta is smooth muscle. My third kid, my wife was in labor 14 hours. She wouldn't have made it. Placenta is smooth muscle. It utilizes energy much differently than a skeletal muscle does to maintain contraction. So this is how smooth muscle evolved. And it has to be that way when you think about it. Just regulating your blood pressure would exhaust you. So we'll talk about that. Smooth muscle is different than other types of muscle, and it can maintain tone. We talk about smooth muscle cell tone. It can generate tension or a resting state of tension called tonus contraction. So your bicep or skeletal, it's off and on. Your cardiac muscle contracts and relaxes. A smooth muscle, whether it's maintaining the lumen of an artery, or if your bladder is filling or you're giving birth, maintains a tonus contraction. It maintains a size or a shape or a certain amount. It's a sliding scale. It doesn't have to be necessarily off and on. Most smooth muscle cell tonic contractions result from prolonged smooth muscle cell excitation. So you can have local paracrine effects on smooth muscle contraction. Okay? Usually circulating hormones. So think about when you get nervous, when you need to go to the restroom. That's local and it's hormonal. Your skeletal and cardiac do not operate that way. So skeletal, I just said this a minute ago, has two states, contraction or relaxation, which is regulated by autonomic mechanisms. Smooth muscle also has a specialized property called stress relaxation. So what is stress relaxation? So I showed you a few slides ago that in cardiac and skeletal muscle, you have sarcomeres, and you have highly organized actin and myosin. Smooth muscle cell, you do not have that. They're kind of randomly distributed, and your actin is attached to an attachment plaque or a dense body. But that's an advantage for the smooth muscle cell. So that smooth muscle cell can rearrange its actin and myosin network at a moment's notice. So this is my really high-tech audiovisual aid. It's a rubber band. But it's not a rubber band. It's a smooth muscle cell. Okay? So if that smooth muscle cell needs to stretch, what's going to happen? If it were any other type of cell, it would snap and break. A smooth muscle cell, since its actin and myosin is really loosely rearranged, it can rearrange itself so that it can stretch without causing damage. Same thing if it's a contraction. It can rearrange its actin and myosin network to fit the need that's necessary at that time. So if you're squeezing materials through your gut, through your lumen, you're doing peristalsis, it can do that. And it can maintain it by organizing its network. So it can change its length greatly without marked changes in tension. So when you stretch out this rubber band, it becomes more tense. But if that were a smooth muscle cell, within a matter of seconds, there's no more tension. And that's really important. So if you think about an athlete, how many ball players tear a muscle or pull a muscle? You don't tear or you don't pull smooth muscle because it can adapt. It can reorganize its actin and myosin network. So in contrast to other muscle types, smooth muscle cell contraction can respond to multiple inputs. So I already alluded to most of these, but there's intrinsic tone. So it's just the molecular makeup of the smooth muscle, and I talked about the actin and myosin network. That helps it sort of maintain a shape. So remember, there's no bone to be attached to. Skeletal muscle's doing this, or you're walking. Your arteries are not attached to a bone. The bladder's not attached to a bone. It needs to have its own intrinsic shape. It can respond to the nervous system via adrenergic receptors, circulating vasoconstrictors and vasodilators, and I gave you a great example last hour when we talked about nitric oxide. There's something called myogenic autoregulation. So just in the stretch of that muscle cell will sort of allow it to compensate and pull back against itself. So it knows when it's being stretched, and it will sort of pull back to maintain its own shape. So think of your bladder filling. Metabolic substances, and again, that's another example, say nitric oxide. So the endothelium will sense when the muscle needs to, smooth muscle needs to relax. So smooth muscle can respond to those types of things. It can respond to other circulating substances in a myocardium. Adenosine will cause smooth muscle to relax. Don't get too hung up on that. And we talked about paracrine substances from the endothelial cells. So the real take-home message from this is smooth muscle cells are bags of fibers that can really rearrange themselves, and they have to because there's no skeleton for them to attach to. They can respond to autonomic signals as well as local signals. Other muscle types cannot respond to local signals. So, quick review, multi-unit and single unit. It's what I saw my own thunder, what I just said. They can respond to either neuromuscular or electrical coupling. And we'll talk about the mechanisms of electrical coupling in a few minutes. Much less energy, velocity is slower, and it's loosely organized. There's some pretty important points. So we have a definition we need to get in our heads, and it's unique to smooth muscle. It's excitation, contraction, coupling. So excitation, contraction, coupling is the process by which excitation triggers an increase in calcium. Of course, calcium is essential for any muscle type to contract or relax. And this is known as excitation, contraction, coupling. So how does the smooth muscle cell get that calcium to raise? So there's two types. The first is called electromechanical coupling. So there's contraction with a change in membrane potential. And I know you learned about membrane potential already. Smooth muscle can generate membrane potential. There's two forms, action potential dependent. However, in a smooth muscle cell, this is much slower. In smoother skeletal muscle, because it depends on calcium channels. Whereas in a cardiac muscle cell, you've already learned all the calcium comes from the SR. And a smooth muscle, the majority of calcium comes from outside the cell and has to come in through a channel. Smooth muscle generates action potentials differently than other cell types. It's not just a snap off and snap on. So there are simple spikes. You can have a plateau and slow wave, depending on where the smooth muscle is, what it's doing. If it's pushing materials through your GI tract, if it's regulating the diameter of a blood vessel, if it's a placenta squeezing a baby out. All different types of waves. So there are two forms of electromechanical coupling. One is action potential dependent, and one is graded depolarization. This causes a lot of confusion with students. A graded depolarization is when a stimulus can cause depolarization without generation of an action potential. It's graded, so you will not get spikes and not get peaks. I'm not going to ask you about this, but I want you to know that it exists. Okay? So try to get this straight in your mind. Electromechanical coupling, there's two types, action potential dependent and graded depolarization. What is the more important and the more unique for a smooth muscle cell is pharmacological coupling. So it's contraction without a change in membrane depolarization. So think of it almost as signal transduction in a smooth muscle cell as a way to elevate calcium. All muscle types need calcium to contract. So this is how a smooth muscle cell will mobilize that calcium. So think about what I said a few times. There's local tissue factors that regulate a smooth muscle to contract. So here's our G protein coupled receptor. They'll recognize that hormone or whatever that peptide is. We have signal transduction. We'll have a second messenger being generated that will allow calcium to come in from the outside. It will also allow some calcium to be released from the ER. But all this takes a little bit of time. It's not as instantaneous as it is in a cardiac myocyte or a skeletal muscle. This takes a few seconds. This mobilized calcium then initiates contraction. So again, let's go back because I've given this lecture a few times in the past. Action potential dependent, that's really not what I want you to really focus on. I want you to focus on the generation of contraction without action potential. That's what's really unique to a smooth muscle cell. And it's really like signal transduction. This is what allows a smooth muscle cell to respond to local hormonal factors. It has receptors to different paracrine signals. You'll get calcium coming from the outside in. You'll have calcium released from the ER, which then activates store-operated calcium channels. The bottom line is you have an increased calcium level that causes contraction. And when we talked about our signal transduction in Block 2, we called calcium a second messenger, and it is a second messenger. It starts up something else. It starts the mechanism of contraction. So this is what you really ought to focus on when you're studying pharmacomechanical coupling. So you're going to get an increase in calcium, but you're not going to get a wave. We're okay with that? So what is the actual mechanism of smooth muscle contraction? They're thick and thin filaments, so actin and myosin, they slide past each other during contraction. So you can read this for yourself, but basically it's an energy-dependent model between actin and myosin, and they walk past each other. So these are some topics we'll talk about for about the next 15 minutes. We'll talk about the rise in calcium, the mechanism about how the smooth muscle cell uses that to contract, the mechanism of excitation contraction, where the calcium comes from. So keep that graph in mind. Smooth muscle cells take longer to generate contraction than the other two muscle types, but it lasts longer. It's more efficient. How we stop contraction, how we can modulate calcium sensitization. I alluded to that in the endothelium lecture. We'll talk about what calcium sensitization is. We'll talk about what a latch bridge is that allows a patch of muscle cells to maintain its shape without using energy. And we'll talk about inflammation and muscle contraction. So keep this in mind. All muscle cells need calcium to contract. It takes longer in a smooth muscle cell because it mobilizes calcium from different sources. It's not as set up to instantaneously contract like other muscle types. So the increase in calcium comes from two sources in a smooth muscle: plasma membrane and the SR. But in the smooth muscle cell, the SR is really poorly developed. In the cardiac myocytes, they're sitting right there. It's right on the sarcomere. It's almost instantaneous. It's more poorly developed. It's more like another cell type. So tension's slower in developing. Because it's slower in developing, it takes longer to reduce that calcium as well, so it can maintain it for longer periods. That allows a slow, steady contraction, so the tonus contraction. And all that is facilitated by that disordered actinomyosin network. They're not arranged in sarcomeres. They have to get themselves together and pull on each other. Calcium falls more slowly. Smooth muscle does not have troponin. It has another muscle called calmodulin. And we'll spend a few minutes talking about calmodulin. So this is a lot of information. What I really want you to keep in mind is this graph. And you have this graph because calcium is mobilized more slowly, and the actinomyosin network is less efficient. But there are benefits to that reduced efficiency as well. Okay, with that? I showed you this slide in signal transduction, except I said it was the currency of signal transduction. It's also the currency of smooth muscle contraction. So years ago, we used to have this lecture in block 2. And they would always try to schedule this lecture before the signal transduction lecture. And I would say, listen, we have to have muscle contraction after signal transduction because it's dependent on kinases, phosphatases. And you'll see why in a minute. So for the same reason, smooth muscle contraction is really dependent on phosphorylation and dephosphorylation for the same reasons. When a protein's phosphorylated, it's different than it was beforehand. It changes shape, it changes activity, it can change interactions. So what I'm going to try to show you when we talk about the actual mechanism of the contraction, when we talked about signal transduction in November, I said it was linear. It went from the outside to the inside. And it started with a change in conformation of a receptor. So the mechanical change was transduced into biochemical change. This is signaling in reverse. We start with biochemistry, and we end up with mechanical movement. And instead of sort of a linear cascade, it's more of a circular cascade. And as we go through the mechanism, you can see. But we need phosphorylation. We need changes in conformation. It's really important to recycle the machinery of how smooth muscle cell contracts. Okay. We're at the biochemical level. It starts with biochemistry and ends up in movement. 5 easy steps. So first of all, we have to have an increase in calcium concentration. And I already said that 3 or 4 times. Comes from the outside, comes from the SR. The SR is less organized, so it takes a while to do it. Smooth muscle has a protein that other muscle types do not have, and it's called calmodulin. Not troponin, calmodulin. In its resting state, it's kind of shaped like a dumbbell. When 4 ions of calcium bind to it, it changes its shape. So it's kind of sort of like phosphorylation. So this calmodulin will change its shape, and it's now a different protein. This is different from this. And it now allows it to bind to another protein called myosin light chain kinase. So in the resting state, myosin light chain kinase is inactive. When calmodulin binds to it, it activates that kinase. And we all remember from those lectures what a kinase does. It puts a phosphate group on a residue, tyrosine, serine, or threonine. And that's what this kinase does. It takes a phosphate and it slaps it on the head of the myosin. So this is a myosin molecule, and this is the head of the myosin. We take an ATP, we result in ADP, and we have a phosphate group on that myosin head. Now that this myosin head has a phosphate group on it, it's different than before it was phosphorylated. Putting a phosphate on there activates that myosin head and turns it into an ATPase, which wants to cleave inorganic phosphate off of that phosphate group. When it does that, the shape of that myosin head changes and it moves. So let's go through this again, and we'll put it in context of how this makes a muscle cell contract. You need calcium for the muscle to contract. Calcium binds to calmodulin. Calmodulin changes its shape. It binds to and activates myosin light chain kinase. Myosin light chain kinase consumes ATP, puts a phosphate group on the head of myosin. It turns that myosin head into an ATPase. It will hydrolyze that phosphate off, and this head moves. Question? Exactly. The next couple slides, hopefully, will make that more clear. So it's going to hydrolyze this off. Because when this is hydrolyzed off, this head is going to move. And I'll put that in perspective in just a second. So I told you that thick and thin filaments will slide past each other, right? So this is actin. This is myosin. This is the head of the myosin. What's going on? So here's our actin. There's our actin. There's our myosin head. So at the resting state, that myosin head is attached to the myosin head is attached to the actin at a 90-degree angle. Okay. When myosin light chain kinase phosphorylates it, it releases and it pivots and it reattaches at a 45-degree angle. So how you can tell that is there's this little green circle here. When it's phosphorylated, it releases from the green circle and binds to this light pink-purple circle right there. But as I told you, when it's phosphorylated, it becomes an ATPase. It will hydrolyze that inorganic phosphate off. In doing so, it releases energy, it changes its conformation, and it snaps back to 90 degrees. When it's doing that, it pushes the actin past it. That's called a power stroke. Eventually, that myosin head has to become dephosphorylated by a phosphatase. Remember, it has to clean the rest of the molecule up. You can't keep phosphorylating the same residue. And it releases from myosin again. So we'll walk through this one more time. Keep in mind what's going on up here with the biochemistry. You have the myosin head at the resting state. You're at 90 degrees. It becomes phosphorylated by myosin light chain kinase. It releases, reattaches at a 45-degree angle. Almost instantly, that myosin head becomes an ATPase. It releases that inorganic phosphate. The energy release causes it to snap back to 90 degrees. And that myosin head will now walk along the actin. So think about if you're in a rowboat and you want to stand still on the rowboat, and we can use my crutches for the oars. You kind of hold yourself there. If you want to move, you have to pick them up at a 45-degree angle and then pull them past you. So when you pull those oars past you, you're hydrolyzing the inorganic phosphate. These are your myosin heads. And you can just row that myosin across that sea of actin, and that's how you get contraction. A lot of students get confused sort of at this last point. I don't want you guys to get bogged down on when this attaches, when it doesn't attach. What I want you to know are these steps. Know the molecules, what happens and why. Know what the power stroke is and how it happens. You have to have phosphorylation of the myosin head. It has to let go, reattach at 45 degrees. It's essential that you know that it's an ATPase. When it hydrolyzes that inorganic phosphate off, you release energy, you snap back to 90 degrees. That's the really important things you should know. When we talk about a latch bridge in about 5 or 10 minutes, we're going to really focus on this last step. It could take 20 minutes to really describe all the proteins that are involved in formation of a latch bridge and that last step. I don't want to do that to anybody. Okay? So get the big picture in your mind, know the players. This is different than how other muscle cell types contract. We're okay? So, as long as you have calcium and as long as you have ATP, that myosin head will walk along that actin fiber over and over and over. And don't forget you've got a phosphatase at the end to clean it up. It will cycle continuously. So you have to have ATP, you have to have calcium, you've got your cross bridge, you've got your power stroke, and that myosin is going to row itself across that actin. So how does that make a smooth muscle cell contract? This is just a molecular model, pre-stroke, post-stroke. So 45 degrees, a little bit more than 90 degrees. So let's look at the cellular level now. This is our smooth muscle cell. And I've said a few times that the actinomyosin network is loosely organized, particularly the actins now. It's really important. The one end of actin is anchored inside the cytoplasm to a dense body. The other end of the actin is anchored inside the plasma membrane to an attachment plaque. So here is our smooth muscle cell that's nice and relaxed, our actin and our myosin. Under the contracted state, when these myosins row themselves across that actin, these will pull together. So here's your relaxed smooth muscle cell, dense bodies and your attachment plaques. When these myosin heads consume ATP and use calcium and row themselves across those actin fibers, since they're attached to the plasma membrane, they'll contract. So think about it. If you have a sheet of smooth muscle, it's in your gut or it's an artery, and you just have a 10 or 15% contraction in the length, and all those smooth muscle cells, since they're unitary, they're in contact with each other through gap junctions, and those calcium ions are flying back and forth in unison. If that whole sheet contracts just a little bit, you can have an appreciable contraction. So you can squeeze materials through the gut. You can have peristalsis. You can regulate blood pressure. You can squeeze that baby out. Okay? So everything is harmonized. Everything is in communication through gap junctions. So we went through the biochemistry, and we went through the cellular level. So this is how it's different than signaling that I described before. It starts with biochemistry. It starts with calcium and kinases, and it ends up in movement. Okay? Probably sick of hearing me talk at this point, but let's review anyway. You need calcium. Calcium binds calmodulin. Calmodulin binds myosin light chain kinase. It's a kinase, does what all good kinases do. Put the phosphate group on the head of myosin. Once myosin is phosphorylated, it's a different protein. It's now an ATPase. Almost immediately, it wants to hydrolyze that phosphate off. When it does that, it changes its position from 90 degrees to 45 degrees. When it hydrolyzes the phosphate group off, it snaps back to 90, and you've just moved a unit. That's called the power stroke. So the myosin heads row themselves along the actin filament. Read it over to yourselves a few times. It'll sink in. If I can get it, you guys can. So, smooth muscle, calcium comes from 3 major pathways. And I've got a nice cartoon on the next slide. So extracellular through voltage-dependent channels, extracellular through calcium-operated channels. So they need calcium to open, to let more calcium in. And intracellular from the SR that's less organized than other muscle types. So because you have to pull calcium in from extracellularly and from these disorganized SRs, it takes longer to increase. So that's why it takes longer for that smooth muscle to pull itself together. Because it does not depend on membrane depolarization, but you have contraction, pharmacomechanical coupling. This is the thing you should really focus on. So this cartoon is what I'm talking about. You get calcium from the outside in a voltage-gated calcium channel. Calcium will do two things. It will release more calcium from the SR. It will also bind to calcium-operated calcium channels. So once it binds calcium, lets more calcium in. It can also activate G protein coupled receptors. Again, why these GPCRs do so many things in our physiology. So there's three major sources of calcium ions. It takes longer to get the calcium level up in a smooth muscle cell than others for that reason. So the party can't go on forever. We have to stop contracting. So if the most important way, or the essential way to increase contraction is by elevating calcium, what do you think is the best way to stop contraction or initiate relaxation? You want to reduce the calcium, right? How do we do that? We can sequester it. We can pump it from the cytoplasm back to the SR, or pump it back out into the extracellular area. And this is all due to the sodium-calcium exchange. And I know you've learned about that. And that depends on membrane potential. So you can get rid of the calcium. That's the best way to do it. So to relax smooth muscle, you require myosin light chain dephosphorylation. So way back when we talked about this, I said you had to keep dephosphorylating that. If you don't dephosphorylate that, it'll stay contracted, right? It'll stay attached. So here's where things get a little complicated. Myosin light chain phosphatase is constitutively active. So it's always taking that phosphate off of the head. Always taking the ADP off of the head of myosin. And this leads to relaxation. There are different ways to tweak this enzyme to maintain contraction. There are different ways to make this enzyme more efficient to sort of increase contraction. So when we talk about calcium sensitization in just a minute, we're going to target the phosphatase or the kinase to allow that cell to get more bang for a buck per unit of calcium. So this is kind of important. So contraction requires the balance between phosphorylation and dephosphorylation of the myosin light chain head. So if you tweak the enzymes that put the phosphate on or take it off, you can sensitize that smooth muscle cell to contraction per unit of calcium. So you can inhibit the phosphatase. That keeps the head on the actin. So you can get more force per calcium. You get more bang for a buck. So you can inhibit the phosphatase. That will cause increased contractility. If you phosphorylate that kinase, it makes it less efficient. It decreases the sensitivity to calcium, and that decreases contractility. So that's one of the things that nitric oxide does. When I said nitric oxide diffused from endothelial cells to smooth muscle cells, I messed around with guanylate cyclase and caused that smooth muscle to relax. This is one of the major reasons how it does it. I want you to just understand the really big picture. You can sensitize the smooth muscle cell to contraction per unit of calcium by messing with the phosphatase and the kinase. If you inhibit the phosphatase, you keep the head of the myosin on the actin, that cell's crunched up. If you make the phosphatase less efficient, it can't rephosphorylate it, so you have more relaxation. We need to talk about latch bridges now. So over and over I've said that smooth muscle can maintain contraction without consuming a lot of ATP. It's because of a unique property of smooth muscle cell called latch bridges. It allows you to maintain high force. So this is called a latch state. So you have your cross bridge cycle. You have your 45 degrees. It hydrolyzes phosphate. It snaps back to 90. But in this situation, it will just stay on that actin. So you've contracted. Instead of treading water or pedaling or doing whatever and phosphatase kinase, phosphatase kinase, phosphatase, it will just lock it right there. This allows the myosin head to hold on to the actin without having to cycle again. And it's this step here that requires so many accessory proteins that we could talk until about 12:15 if we really want to get it right. Let's really keep it simple. I want you to know what a latch bridge does and how it allows maintenance of high force with low energy. You don't need an ATP to keep phosphorylating this every time. That's how we can maintain our blood pressure when we're unconscious, how, we don't have to run to the restroom every 5 minutes. You just kind of latch onto it and hold onto it. That's what, you can't do that in a skeletal muscle. That's why you feel fatigue after about a minute of trying to do this. We're okay? We need to talk about this protein called caldesmin. Caldesmin is also unique in smooth muscle, and it sort of links inflammation and links other processes with maintenance or with regulation of contraction. Here's what caldesmin does. It binds to actin when there's low calcium. So in the relaxed state, caldesmin binds to actin. You have steric hindrance. It gets in the way of the myosin head. Smooth muscle is relaxed. Perfect. When caldesmin becomes phosphorylated, the affinity for actin reduces and it goes away. Phosphate should go away with it. But it goes away. It allows the myosin head to interact with the actin and it allows contraction to proceed. Why am I telling you all that? So you remember this figure from our signal transduction lecture, our old buddy MAP kinase. MAP kinase is what phosphorylates caldesmin. So if you have a growth factor receptor that's activated, if there are cytokines or any kind of inflammation around, or even if it's a receptor for a local factor, a local hormone, I said smooth muscle cells can be activated by local factors. MAP kinase will phosphorylate caldesmin, make it go away, and increase contraction. So this is another mechanism of how a sheet of smooth muscle can respond to local factors, whereas skeletal and cardiac cannot. It's also a way that when you have some low-level localized inflammation, you can have increased contraction. Now that's a big deal if we're talking about hypertension, chronic high blood pressure. So if you have a lot of vascular inflammation, you have athero, you also tend to have hypertension along with it. And this is one of the mechanisms how that happens. So I just talked a lot. I just want you to know what caldesmin does. It binds to actin. It prevents myosin from binding. It goes away when it's phosphorylated. So this is another way of presenting the nitric oxide that I showed you in the endothelial lecture, but here's what kind of ties it all together. We have paracrine signaling from the endothelial cell. Nitric oxide's a gas and diffuses. Guanylate cyclase, I said, causes the smooth muscle to relax by changing the calcium sensitivity. So it decreases the intracellular calcium. It helps it sequester away. It also changes the myosin light chain phosphatase. So this is really the mechanism. Don't get hung up on terminology. I want you to know where the nitric oxide comes from. It changes calcium sensitivity in the smooth muscle cell. And you know what calcium sensitivity is regulated by, phosphatases and kinases. And I showed you this picture before. So now you know it. In here you've got actin, you've got myosin. When the myosin head's phosphorylated, you have the whole process of contraction. But if you have nitric oxide in there, it's going to decrease the sensitivity of that kinase per unit of calcium. You'll have less contraction. We're done. Okay, so let's go through this. I threw a lot at you. I tried to do my best to emphasize what I think is really important. Don't get bogged down with minutia. Keep the big pictures in your mind, who the players are. Smooth muscle is different from other muscle types because of disorganized actin and myosin filaments. Know that the actin is attached on the outside of the cell as well as on the inside of the plasma membrane. No muscle tone. What stress relaxation is, our buddy, the rubber band, you can stretch it without increasing tension. And that's because actin and myosin can deregulate its organization. Multiple inputs that allow smooth muscles to contract or relax. Many different inputs. There's electromechanical coupling and pharmacomechanical coupling. You know I spent about 5 or 6 or 7 slides on pharmacomechanical coupling. For a smooth muscle cell, I think that's more important. We spent a lot of time talking about the mechanisms of smooth muscle contraction. You have to have the rise in calcium. Calmodulin changes its shape, activates myosin light chain kinase, phosphorylate the myosin head. It hydrolyzes an organic phosphate. It releases from the actin, reattaches at 45 degrees, hydrolyzes it off. You snap back to 90. You just move the unit. You can't keep phosphorylating that myosin head. You have to have the phosphatase to remove the residue and start over again. And this will be about the 3rd or 4th time we're talking about nitric oxide and how it works. It tweaks with the phosphatases and the kinases and that smooth muscle cell that regulate contraction. So read through this a few times before you panic that you don't understand it. It's really straightforward. I'd be happy to answer any questions if you have them. But we're done. Whoop, wrong way.
Hey, Woodley. Hey, Doug, how do I turn the do you know how to turn the well, the projector's not on.
There. All right, let's see. I think
It is PC, right? Supposed to be PC.
it should be PC, and then
PC first.
display on stage left projector. It's not that.
Wait, I really. Is there any students who
Wait,
know
how
how to
about
do
this one? Stage left projector
I
picture
did that
mute.
mute.
That doesn't unmute it.
I think it's got to be.
Wait, projector is warming. It looks like it might just need time. There it is. It's coming
okay,
up.
Okay. Thank you. Do you want this, or you just want to talk loud?
Well, you're doing the main thing, so
No, but you can have this, and I can have this.
all right, leave it up here. I'll put it on it because otherwise, when I make comments or something.
All right. Yeah. Yeah, that's right. That's right.
Well, I was in St. Parks for a week. I've been back for a week. So.
Okay, guys, let's get started. So thank you all for coming. So this is a regular town hall. We'd like to do them about twice a year. We'd like to really sort of follow the same agenda. So it's a town hall related to how we're best representing our health, equity, diversity, and inclusion mission, which is really very important to Dr. Kaiser and to the leadership of our school. So what I'd like to do is sort of review where we are, just in the first 20 minutes or so, of the things that were brought up in the previous town halls, where we are with the diversity strategic plan, and talk just quickly about some of the other new things that are happening, and then leave 20 to 30 minutes left for you guys to bring up whatever issues or questions or concerns or good things that you have with Dr. Kaiser here to give some input. So the information here is taken from the town halls both last November and last February, and the issues that you brought up and what we've done to date to try to address them. So a big issue, of course, has been recruitment and retention of underrepresented students, residents, and faculty members. So under Dr. Kaiser's leadership, the admissions committee has been completely restructured. The dean of admissions is no longer the chair of the committee. A number of underrepresented faculty members were added, as well as Dr. Cosby and Dr. Arasarina are on the admissions committee. So the face looks different. Our students are right now primarily underrepresented students and are providing great input to the committee. We're actually doing unconscious bias training for the admissions committee on Friday. We've talked about really meeting our mission of primarily African American, Black, and Hispanic students, as well as educationally and economically disadvantaged students. Hispanic is really grade aided. Students from Puerto Rico are really students we're primarily recruiting, and then students from Central America, and then students from South America. Spain and Portugal, again, aren't going to be included in our plan. We're looking at really students who meet our mission. And regarding faculty, there's a new faculty hiring process. with Mike Lester, who runs physician recruiting. So our office now is very involved. We never had a faculty hiring process policy before, so we have one now. There is a member of the Diversity Council on each search committee. Every search committee will go through unconscious bias training, and there will be no more selected hires. So I think all of those are really positive things. And our number of underrepresented faculty hires over the past year, it's still way too low, but it doubled. So we're at least moving forward. And of course, finances is such a big, important issue. So Dr. Kaiser was able to commit half of all of our scholarship dollars given to recruitment to underrepresented students, and even found some other funds.
Yeah, we added $2 million to scholarships. What, just a few months ago?
Yeah.
Just in the middle of the year, we were able to add $2 million to scholarships. So all good.
And we completed the hiring process with Dr. Arasarina. Students were involved in that from start to finish, so I think that really improved the process. Our office is fully functioning, and I think speaking to the importance to the institution, we've moved over to the 3rd floor, so we're really in the center. I think it's really helped our interactions with the other dean's offices and given people great access to the folks in our office.
Yeah, I think being able to relocate the office to be more in proximity with the other student offices, I think was I think something that people really should take advantage of. You no longer have to go find the office somewhere. It's centrally located now.
Yes, where's Kresge? There was also an identification of the need for really better structured academic support for underrepresented students. So, that this takes money. So we're very fortunate that Dr. Kaiser gave us finances to be able to provide a summer enrichment course specifically dedicated to underrepresented students. The PrEP program, which I think many of you took part in, has been really successful. It's a 3-week program prior to starting medical school for students who are underrepresented or students who have a reason that they feel concerned about their ability to assimilate into medical school. And it's really gone quite well and resulted in a strong mentoring program. And other resources for some extra URIM support for some of our students. We also talked a lot about LGBT issues. There's a new LGBT task force. It was started by faculty, by Rob Bedecker and Heather Klaus. But we're working hard to provide our offices support, and we're going to look with the new year moving forward how to provide more support to that venture. They've done a great job with that. We've had a lot of discussions with admissions. There was a movement to get information to applicants about the LGBT community, and now that's available in the lobby during the admissions process, but will be part of the admissions brochure next year. We're also looking at redoing the supplemental application